Alright guys, so we are going to be moving on to our next to last unit. We have pretty much the kingdoms left to cover, and that's going to involve prokaryotes, and we're going to talk a little bit about protists and fungi, and that's going to be what this unit that we're doing right now is going to comprise. And then our final unit is going to be a combination of plants and animals. We're actually going to group those together into just one bigger unit instead of breaking it up. Because every time we break things up, we have to have more days for review and test, and at some point we're just going to run out of days. I know you guys are probably all getting tired, and that senioritis and junioritis is definitely setting in, but I just want to encourage you to keep pushing forward. We just have a little bit more to go. So we just finished up our evolution unit, and one of the things we covered in evolution was taxonomy, and we talked about how organisms are grouped. And so essentially what we're going to be looking at are some of the major taxonomic groups and how they're broken up and their characteristics. And like I said, this is going to take us through the rest of the year. So we're going to start off with prokaryotes today, and I would say about half of the information that we're going to cover is probably going to be review, okay? So for prokaryotes, we've already talked about them back in first semester when we did prokaryote versus eukaryote. We actually talked about prokaryotic genomes when we covered DNA technology, and we talked about things like plasmids and their chromosome. And some of this you just may know from just basic common knowledge. So as we move through the prokaryote notes, like I said, some of this you're going to recognize, some is going to be new. Now, when it comes to prokaryotes, remember your prokaryotes are your cells that do not have a nucleus. They do not have the membrane-bound organelles, and they are everywhere. Okay, this little fun factoid here, to me, is really kind of hard to believe. I believe it, but it's kind of crazy to think about, that if you took their collective biomass, now keep in mind we can't see prokaryotes, they are microscopic, they are unicellular, but if we took all of the prokaryotes on Earth, the mass of those prokaryotes would be greater than all of the eukaryotes. And so when you think about eukaryotes like trees or elephants or even humans, I mean, things that have so much more mass as an individual, the prokaryotes outweigh all of us. And that's because there are just simply so many, okay? Prokaryotes outnumber everything. They are found everywhere. Okay, if life is possible, you will find prokaryotes there. In fact, there's going to be places on the planet where probably only prokaryotes exist because eukaryotes have not been able to survive there. So we have about 5,000 known species, but look at the estimate. 400,000 to 4 million species are probably out there because they're in places that we really can't even study and visit. And, of course, they're microscopic, so we can't easily see them with the naked eye. Now, we have covered this before. We have, in terms of taxonomy, we have two domains, domain bacteria, which includes kingdom bacteria, and domain archaea, which includes kingdom archaea. Now, I do want to mention something really quickly because depending on the source that you're looking at, it wasn't but just a few years ago that when I was teaching this, these kingdom names were different, okay? So sometimes on certain resources, in fact, you may have worksheets or you may have activities that you do even this year where you see the terms eubacteria, EU bacteria, and archaebacteria. Because for a time, the kingdom names were eubacteria and archaebacteria, which means true bacteria, and then you had the archaea prefix. Now, as far as I know, because taxonomy is something that is constantly changing, but as far as I know, these are the accepted kingdom names, okay? So you may have learned something different back freshman year. You should have learned bacteria and archaea, but I know it was probably around that time, especially for you seniors, that some of these terms were changing. So for now, what you need to know is the kingdom and domain names are the same, okay? And we're going to go into their characteristics and what sets them apart, but just know that bacteria or go with bacteria and archaea go with archaea, okay? But if you ever come across the eubacteria and the archaebacteria, those are just some former names for those kingdoms. And so a couple of things to show you. Here's just a very simple cladogram, okay? On this one you can see that it's believed that domain eukarya and domain archaea came off of the same branch, okay? Here's another one that kind of breaks it up into some of the larger groups. And we're going to take a brief look at some of these groups and what they are. But you can see that, once again, domain bacteria, there's several different types. But here we have the branch, and both eukaryotes and archaeans come off of the same branch. So we're going to start with domain bacteria. These are the things that we commonly call bacteria. Okay, they are very diverse. One of the key characteristics, and we've learned this before, is that they have a substance called peptidoglycan in their cell walls. This right here is going to be one of the big separators from bacteria and archaea, is the presence of peptidoglycan. And pretty much all it is, peptido, that pep tells you there's probably some proteins. Glyc is a prefix that tells you there's some sugars. So this is going to be like a protein carbohydrate type of matrix, and that's what they have in their cell walls. So as we go through these kingdoms, as we go through taxonomy, we're going to see how important the cell wall is in classification. Because when we get to fungi, they're going to have something different. When we get to plants, they're going to have something different. And when we get to animals, they're not going to have one at all. So the cell wall is a very important feature in classification. Okay, so for bacteria, bacteria have PEP. Okay, and like I said, these are your common everyday bacteria. The ones that we were probably going to come in contact with, the good ones, the bad ones, the ones that may live inside of us or on us. E. coli, which is both good and bad, it depends on the strain. You've probably heard of streptococcus before, that's what causes strep throat. Staphylococcus, you've probably heard of a staph infection. So many different bacteria that we come into contact in our daily lives. Now here are some major groups. I know this is a lot to remember. Some of these are pretty self-explanatory, okay? I definitely want to highlight this one. If I were you, I would star that one. This one is very significant because of evolution. So we talked about something known as endosymbiotic theory. And the evidence for the endosymbiotic theory are chloroplast and mitochondria. It's believed that prokaryotes were engulfed by other prokaryotes, and that's what kind of gave rise to these internal membrane compartments, or these very complex organelles like the mitochondria and the chloroplast. So cyanobacteria are actually photosynthetic bacteria, and they're believed to have been the ancestors to our modern day chloroplast. Remember that those chloroplasts and mitochondria, they have a double membrane which supports that engulfing. They have their own DNA like a prokaryote does, their own ribosomes, their own enzymes. They're like little cells within our cells. And so these are believed to be the ancestors to those chloroplasts that you find in like plants and protists. Now some other groups that you might be familiar with, like I said, you don't have to necessarily know all the details. But proteobacteria, these are a lot of gram-negative bacteria. I know we probably don't know what that is just yet, but we're about to talk about it. E. coli fits in this category. Chlamydias, that may sound familiar because chlamydia is a common STD. So I want you to notice that these are parasites, okay? Spirochetes, here's another STD, syphilis, as well as Lyme disease that can be carried by ticks. So the spirochetes get their name because they're spiral shaped. And then gram-positive bacteria, once again, we haven't really talked about what that is, but your basic strep and staph would be in that category. So for quiz purposes, just kind of know the general definitions, okay? But once again, I would definitely focus on that one. That one's pretty significant, okay? All right, so for domain archaea, these are your extreme prokaryotes. Technically, we really should not use the word bacteria when we're talking about archaeans, but they are going to be prokaryotes that are found in extreme locations. So they're going to be in places that a lot of other living things would struggle to survive in. Things that are very hot, things that are very salty, things that have no oxygen, places that are very acidic. So they are extremophiles, okay? Methanogens, they live in areas where there's no oxygen. Halophiles and thermophiles. Halophiles, I think, live in very salty environments. Thermophiles live in very extreme temperature. But one of the key identifiers here is they're prokaryotes that do not have peptidoglycan. Okay, so that right there is one of the key separators. Bacteria have PEP, archaeans don't. Now, it's not that they don't have cell walls necessarily, it's just because they are so diverse and extreme, they're going to have specific substances that make up their various cell walls. They also have some unique lipids in their membrane. And so I don't really know what makes up their cell walls because they will vary from Archaean to Archaean. And as we saw with our little phylogenetic trees, they're believed to have just as much in common with eukaryotes in terms of studying some of their DNA and their RNA as they do to the other prokaryotic groups. All right, and here we have 4 major groups of Archaeans. How they even discover some of these, I have no idea. But you have your core archaeotes. They live in hot springs, so they're one of your thermophiles. Euryarchaeotes are going to be some that are anaerobic. They don't use oxygen. They actually reduce CO2 and produce methane, which is where they get the word methanogens. You have crenarchaeotes. A lot of these live in extreme temperature or extreme pH. And then you have nanoarchaeotes, which these are a parasite that grow or that live off of the crenarchaeotes. And so the hydrothermal vents are where you can find those. That's definitely an extreme environment on the bottom of the ocean floor. Once again, just kind of know some general characteristics. You may have a couple of questions on a quiz or a test, but there's not going to be a lot of focus put on these major groups. All right, so we're going to move forward and talk about structure. So most of them are unicellular. They're one cell. And so they may colonize, and what that means is they live together in a colony. They're not necessarily one organism, but they might live in close proximity. There's 3 basic shapes, okay, and you're going to be using these in the lab that you're going to do, especially when we're talking about like diseases, which is what you're going to be researching. But in terms of classification of where do these bacteria go, one way that they're going to be classified could be based on their shape. And so we have cocci, bacilli, and spirilla. And so cocci are spheres or circles or they're round. And the way I like to remember that is, see, you've got the letters C, O, C, C. Those are all very round circular letters. And so these are going to be circular and round. Now sometimes they're single. Sometimes they may be paired up like that. It's called a diplococci. They could be in a colony where there's several of them kind of clumped together. And like we saw earlier, Staphylococcus and Streptococcus, those are some examples of some cocci bacteria. And so that's one of the things they're going to use to identify like bacterial infections is by looking at their shape. Bacilli are rods, and so here we have the letters I, L, L, I. And so those are kind of rod shaped. They're kind of more oval. I don't know if you guys have ever heard of the game Dr. Mario, but anytime I see bacilli, it makes me think of the little peels that you see in Dr. Mario. And then you have spirilla, which that name's kind of self-explanatory. They are spiral or helical shaped. All right, another way we classify them is based on those cell walls. Okay, so as we saw earlier, bacteria have peptidoglycan, Archaeans do not, so that's one way that we can distinguish the 2 kingdoms. But if we have peptidoglycan, okay, so if we're talking about bacteria, we can identify them by a gram stain. And this is where we get the terms gram positive and gram negative. So this is based on the amount of peptidoglycan. Okay, so you have to understand that a gram stain is only used to identify bacteria. A gram stain would not be helpful for Archaeans because they do not have the peptidoglycan. So depending on the amount, you either have a large amount of pep or you have less, okay? And so based on the amount of peptidoglycan, these bacteria will stain differently in this process known as a gram stain. So if they have really thick peptidoglycan cell walls, they are going to stain purple, okay? And so we call those gram positive. And so the way I remember this is positive, purple, lots of pep. Okay, positive or purple, lots of pep. For me, purple's my favorite color, so that's definitely a positive thing. And so positive, purple, lots of pep, okay? Now, if they do not have a lot of peptidoglycan, in fact, a lot of times they have an extra layer around their cell wall. And so what happens is that purple dye rinses out. So when they come back with the second stain, it actually stains a reddish color, okay? Sometimes it's kind of pink, but I like to use the word red. So gram negative, red, less pep. Now, red often has a negative connotation. If you think about blood, blood could be a negative thing if you are bleeding. If your business is in the red, that means you're not making money, you're in the negative. So I try to associate just some simple word association. So positive, purple, lots of pep; negative, red, less pep. And so we can pretty much, if you're talking about bacteria, with a few exceptions I'm sure, you can either group your bacteria into either a gram positive or a gram negative. And as we saw earlier in those groups that we were looking at, a lot of them were grouped based on whether they were gram positive or gram negative. So here's an example. If you take microbiology in the future, you'll probably definitely be doing some gram stains. But here's an example of a gram-positive cocci bacteria. So you can see that it's round in shape. It's stained purple. So this is gram-positive cocci. Here we have a gram-negative bacilli. We can see the rod-shaped cells. It's stained red, so that would be a gram-negative. It's kind of like saying a human has brown hair, blue eyes. Okay, this is the way we describe your bacteria. Now, some other things you might find with bacteria is they might secrete what's called a capsule, and that kind of helps them better invade and kind of attach to other tissues. Or they might have little structures called pili. Back in the very first semester when we were covering cells, you had to label a prokaryote on your little lab activity. And that prokaryote had these little pili. I believe they also call them fimbriae. But these pili are for sticking, okay? These pili are not for moving. And I want to point that out because pili look very similar to cilia on a eukaryote. Now, I'm not going to say that it doesn't exist, but I have never heard of a bacteria having cilia, okay? As far as I know, cilia is a eukaryote thing, okay? Now, both prokaryotes and eukaryotes can have flagella. But when it comes to cilia, cilia are hair-like structures for movement that I've only ever seen on a eukaryote. These pili have a very similar appearance. They kind of look like hair-like structures, but they're not used to move, they're used to stick. Okay, so pili are used to stick to other things. Some of your bacteria can create what's called a biofilm, and if you have teeth or if you have a retainer, you have experienced biofilms because plaque is a biofilm that forms on your teeth. So what happens is these bacterial communities, they secrete this really sticky, sugary type matrix, and it traps cells in it. And it's very, very, very strong. It's very difficult to kill, and it's very hard to get off. It can form on your contact lenses. It can form on like artificial parts that they put like in replacements, or if they put any kind of hardware inside to fix something like bones. It can also form on your teeth. Like I said, that's plaque. I know this is kind of gross, but I have one of those clear retainers, and there's stuff on it. It's like this whitish color material. It doesn't matter how hard I scrub with the toothbrush, that's not coming off. And I'm almost positive that that is a biofilm. Something else that I noticed is back in COVID time when we could wear those clear like mask screens in front of your face, kind of like a shield. I noticed that where my breath would hit, there would be stuff on that shield and it would not wipe off. And so these are pretty common. They are difficult to remove. And if you think about the plaque on your teeth, pretty much you have to scrape it off. When you go to the dentist, they're scraping it off with those metal instruments. It does not come off easily with just a basic toothbrush. That's one of the reasons we need to brush our teeth, is to kind of prevent some of these biofilms from forming in the first place. All right, so now we're going to move on to motility. So this is how the bacteria or the other prokaryotes can move around. Some have flagella. Now, like I mentioned earlier, both prokaryotes and eukaryotes can have flagella. They are tail-like structures that kind of help them move. Now, the structure is a little bit different. In a eukaryote, the flagella is going to be a lot thicker and it's surrounded by the cell membrane. But in bacteria or other prokaryotes, they are much thinner and they're not surrounded by that membrane. So same basic concept, but a little bit different in structure. They could have one or several, it just depends on the bacteria type. Some of them, like those spirilla bacteria, they can move in a corkscrew-like movement. They kind of are able to bore down into tissues. It sounds kind of disturbing, but that's how they kind of infect. Some of them will secrete a slime and then kind of move along it. And then some don't have really a directional type of movement. They just kind of maybe move randomly within whatever substance they are in. They don't necessarily have a particular direction. All right, and that takes us to taxus, which some of the things we're going to talk about in this set of notes are not only referring to, they don't only refer to bacteria or prokaryotes. So taxus is something that pretty much almost any living thing can demonstrate. But even these unicellular prokaryotes demonstrate taxus. And so this is going to be the movement toward or away from a stimulus. So if it's a positive taxus, they're moving toward it, like they may move toward food. If it's a negative taxus, they're moving away from it, like they may move away from chemicals. They could move toward or away from light. And then we see these prefixes, chemo and photo. Okay, chemo always refers to chemicals, photo always refers to light. So these are just going to be responses to light. And once again, these terms are not limited to prokaryotes. Okay, animals can demonstrate both chemo and phototaxis as well. All right, the genome. Now this is the material that we've actually covered before. Remember that anytime we see the word genome, we are talking about the genetic instruction or the DNA. So they have about 1/1000th of the DNA of a eukaryote. They do not have nearly as many genes, so it's a much simpler, smaller genome. They do not have a nucleus, so their DNA is in what's called the nucleoid region. Now their DNA is double-stranded, just like ours is. All DNA in living things is going to be a double-stranded DNA. What's different is the chromosome structure. So where we have those chromosomes that look like X's, okay, prokaryotes have chromosomes that look like a circle. All right, but it's still the same double-stranded, double helical structure of DNA. Now, as we saw in DNA technology, one thing that's common with your prokaryotes is they often have these little extra pieces of genetic material called plasmids. They're just small circles. They don't have to have them to survive, but they may give them some additional genes and traits. All right, we've seen this before as well. They reproduce by binary fission. It's essentially copying their DNA and dividing in two. We do get identical cells, so through reproduction alone, they're not really generating a lot of diversity. However, they can get some genetic diversity through gene transfer, and we've seen these three before as well. All right, transformation is when they take up genes from their surroundings. I'm not sure why that picture is blocking that there. But they just randomly can take up genetic information from their surroundings. One of the labs that we kind of did virtually was a transformation lab. And so it's all about getting a bacteria to take in a gene from their surroundings. They get them to take in the plasmid. Conjugation is when they exchange those plasmids through a sex pilus. They literally kind of join up and they exchange a plasmid or plasmids through a hollow tube called a pili. It's just a specialized pilus. And then transduction is when viruses may transfer genes from one bacteria to another. Now the main way they can generate diversity is just through random mutation, but they could also generate some diversity through gene transfer. And so once again, we've talked about all three of these before. All right, the nutrition and metabolism. This is another set of terms that this applies to all living things, okay? This is the first group that we're talking about in terms of taxonomy, but as we go through, every other group is going to fit in one of these four categories or multiple, okay? Now, the reason we're talking about it here is because prokaryotes are so diverse that you're going to see examples of prokaryotes in each of these four categories. Now, if you've paid attention over the year, you should already know what these words are. Okay, so we're going to start with the root words. We have autotrophs and we have heterotrophs. So let's go back to our ecology lesson. And an autotroph is anything that makes its own food. A heterotroph is anything that cannot make its own food and therefore has to get energy or food from somewhere else. Okay. So now we take a look at the prefixes, and here once again we see photo and chemo, photo and chemo. So photo is light, chemo is chemicals. So if you have an autotroph that's a photoautotroph, we know that it's making its own food. How? It's getting energy from light. So this is essentially going to be your photosynthetic organisms. This is going to be your plants. This would be those cyanobacteria that we mentioned earlier. Okay, chemoautotrophs are going to be organisms that can make their own food, but instead they're using what they need from chemicals. Okay, they're using energy from chemicals in order to produce sugars. So you're going to find chemoautotrophs in places where there's no sunlight, maybe in a bottom of a cave, bottom of the ocean, places like that. Now, photoheterotrophs, these are kind of a weird group. I don't really know of a lot of specific examples of these guys, but they do have to consume organic compounds. They can't make sugars like glucose, but they are able to derive some energy from light, okay? And then the last group, your chemoheterotrophs, they're going to have to consume. They're going to be reliant upon eating for both energy and organic compounds. And so chemoheterotrophs would be like what we are, okay? We pretty much rely on the food that we eat for all energy needs. All right, next set of terms, metabolism and oxygen. So how do these organisms take the food and turn it into energy, okay? And so once again, we have some vocabulary that we should recognize. We're just going to add to it a little bit. So we have aerobes versus anaerobes. Now this goes back to aerobic versus anaerobic respiration. Aerobic means it uses oxygen. This is cellular respiration. Anaerobic means it does not use oxygen. It is fermentation, okay? So now let's take a look at the words that we have in front. We have obligate and facultative, okay? Now, if you are obligated to do something, that means you kind of have to, right? If you are obligated to go somewhere with your parents, you really don't have an option. You have to do what they say, right? So obligate aerobes are obligated to oxygen. So pretty much the only way that they're generating ATP is through cellular respiration, which is aerobic respiration. They have to have oxygen to survive and make ATP. Now I'm going to jump down to obligate anaerobes really quickly. Now they're obligated to no oxygen. In fact, they are going to be poisoned by oxygen. They're going to live in areas that are not oxygen rich. So like those methanogens, okay? And what they're going to do is they're going to live exclusively by fermentation. Now they aren't going to be making a lot of energy, but they can't be really in the presence of oxygen. So they're going to live in places where really no oxygen is there, like maybe kind of like in the mud and muck, or they could live in areas like even inside of another organism, but they're going to only rely on anaerobic respiration. Now, a facultative anaerobe can do both, okay? Facultative means to kind of help. That's really not helpful in this particular scenario, but they aren't obligated to one way, okay? Now, oxygen yields more energy, so it's always better to do aerobic if you can. And so as long as oxygen's present, they'll use the aerobic pathway, but if for some reason oxygen's not, they can also follow that anaerobic pathway. So these guys are able to do both. All right, and to me, one of the most important parts of everything that we're going to talk about from here on out is what is their ecological significance? Like, why do we need them? What do they do? And what you're going to find is that prokaryotes have a lot of different roles. You know, a lot of times when we talk about bacteria, our first response is kind of, ooh, gross, and we want to get, some hand wash, or we want to get stuff that's going to get rid of the germs. But the reality is, we would not be able to survive without bacteria. There are a lot of very positive roles of prokaryotes. So they can be decomposers, which we know is an essential component to any ecosystem. They could be producers, producing food for living organisms and producing oxygen. All right, they're very important in those nutrient cycles that we looked at. We talked about nitrogen fixing bacteria. We talked about denitrifying bacteria. Those decomposers could be involved in those nutrient cycles. They're also helping recycle and move carbon dioxide throughout the atmosphere and Earth. They are often part of symbiotic relationships. Many of them are mutualistic. Like we have gut bacteria that help us digest and help us produce vitamins. We have bacteria that live on our skin and help kind of maintain and keep bad things away from us. Now, there are parasitic bacteria, and these are the ones that are going to give the bad names, but many of them are good bacteria, and we'd be in trouble if we didn't have them. They could be pathogens, and like I said, this is what can give them the bad rap. Some of the diseases can be very, very bad, and this is what we're going to focus on in our kind of research lab that you're going to be doing. But pathogens just are any disease-causing agent, and obviously bacteria can cause disease. Now, if they are causing disease, they're usually either doing one of two things. They're producing endotoxins that when they're broken down, they release these chemicals. A lot of your food poisonings like Salmonella and E. coli, they release these endotoxins. Or some just secrete an exotoxin like tetanus, cholera, plague, botulism. All of those are going to be a type of exotoxin. But pretty much what's happening is either they are breaking down your tissues for food. That's normally what's happening, or they're literally eating your tissues, and so that's how they can be disease causing, which is going to trigger immune responses and definitely damage some of our tissues. So we're familiar with the fact that if we don't treat bacterial infections, it can be deadly. So they definitely can be quite harmful. Luckily these days we have antibiotics to kind of help us out, but they can definitely harm us for sure. All right, so Koch's postulates. There used to be an essay about this. We don't have one anymore, but you can still see a question about this. So how do we know a particular bacteria causes a particular disease? And so this is kind of a set of rules that are followed. So like, how do we know Strep pyogenes causes strep throat? Okay. Or how do we know that Heliobacterium pylori, or whatever it's called, causes ulcers? I think it's Heliobacter pylori. But like, how do we associate a particular bacteria with a particular disease? Well, it has to meet these criteria. So number one, every time a person has that disease, that bacteria must be present. Okay, so we know that if you go to the doctor, a lot of times they take swabs and they culture. They try to figure out what bacteria do you have. That bacteria must be cultured from a sick host. So first, it has to be present. It has to be cultured. Then what they'll do is they'll take those bacteria and they'll put it in another host, okay? Now, obviously, we wouldn't do this with humans, but like with something like rats, something like that in a lab. So they take that bacteria that was cultured and then they put it into a new host, and it must induce the disease, and then they must be able to recover that bacteria from that newly infected individual. So the bacteria has to be present. We culture it from a sick host. We put those cultured bacteria into a healthy host where it must cause the disease again, and then it must be recovered once again. And so if these rules are followed, we are then able to say that a particular bacteria caused a particular disease. And then some other things that we can use bacteria for, they're definitely used heavily in research. We know that prokaryotes, we saw this a lot with DNA technology and genetic engineering. We can use them for bioremediation. In the video you watched called Playing God, the guy was getting the patent on bacteria that can clean up oil spills. We use bacteria in our sewage treatment processes. We use them to make commercial products, and we use them in food, like to make yogurts. So we actually use them quite a bit in our everyday lives. And that is going to wrap up our prokaryotes. So that is going to be our first two taxonomic groups. We have domain bacteria and domain archaea. And we're going to be moving forward with protists and fungi in our next lesson. And so I do want to go ahead and take just a minute to introduce you to the lab that you're going to be doing throughout this unit. So this is what it looks like. Okay, you're probably going to be getting this today. And I just want to point out kind of what we're looking at here. So first off, this whole thing is going to be based off of research. Okay, all of these questions are going to be things that you're going to research. You're just going to use Google to answer. And then the pictures we will address at a later time. I'm going to help you out with those. But what I want you to focus on right now is only the first page. Okay, so notice this is protist here. The last portion is about fungi. So the only page I need you focusing on right now is the prokaryote section. And so the combination of today and tomorrow, you're going to be working on this in class. And so what you're going to do is you're going to research these bacteria. Okay, so these are their scientific names. You can see based on the format, capital letter, lowercase letter, in italics. This is the genus and species. And so your job is going to be to research and identify their gram stain and shape, like it could be gram-positive cocci or gram-negative bacilli. There is one that's not necessarily for sure a positive or a negative. They usually classify it as a negative. But you're going to find out that general information, and then you're going to figure out what disease they cause. Now, you can be as specific or general as you want to, but on a quiz, you will be expected to match some of these up with their description. And that description is going to include the stain, the shape, as well as the disease and the symptoms. So you're just going to find that by research. Now, we actually have microscope slides for just about all of these, if not all of these. But I'm going to be honest with you, when you look up microscope slides for bacteria, they're still incredibly tiny. So what I want you to do is just to Google a picture of what gram-positive cocci would look like. You probably want to include color because gram-positive and gram-negative are represented by colors. So here you're just going to Google and draw a picture of what those would look like under the microscope. And that's the only section you need to focus on right now. Okay, we'll come back to the protists and fungi at a later date. And that is all that I have for you today. I hope you guys had a great little break, and I will hopefully be there soon to kind of check in with you, and hope you guys have a great day.